Welcome to Get Real Health. I'm your host, Dr. Chana Davis. This show cuts through the noise to give you science-based insights from real experts so that you can make smart, healthy choices. My guest today is Dr. Bronwyn MacInnis. We'll be chatting about COVID-19 evolution, transmission, and diagnostics. We'll be tackling some of these media stories around strains and the differences between them. We'll talk about diagnostic capacity, what we have and what we need to get things under control. Dr. MacInnis has had a multi-decade career in the field of genomic epidemiology, which means using DNA and RNA to track how viruses and other pathogens spread and change throughout an outbreak. In the past, she's worked on Zika virus, Ebola virus, and malaria. She's also done a lot of work on bringing diagnostic and genomic testing capacities to low-resource parts of the globe. Dr. MacInnis is an associate director of malaria and viral genomics at the Broad Institute, a partnership between MIT and Harvard University. She's a visiting scientist as well at the Harvard School of Public Health. Dr. MacInnis did her PhD at the University of Alberta. She did her postdoctoral studies at Stanford University, and that's where I'm really glad I bumped into her. She and I have been fast friends for the last 15 years, and even to the point where she's the inspiration for one of my daughter's names. So it's a real pleasure, both professionally and personally, to speak with Dr. MacInnis today. I'm so thrilled that she was able to carve out some time because she is really, truly in the trenches with COVID-19 research. Dr. Bronwyn MacInnis, thank you so much for taking the time to talk today. I know your plate is extremely full, so this time is really appreciated. We have a lot I want to cover in roughly 30 minutes, so let's just dive right in. I guess the big three areas we'll talk about will be strains and sort of evolution of the virus. Second one being viral transmission and sort of dynamics, and the last one will be diagnostics. So we'll just jump in with the strains and the evolution part of it, because that's one I think is getting a lot of media, and there's a lot of sort of confusion and misinformation out there. So the big question I want you to answer is, what's actually happening in terms of how the virus has changed since it entered humans.
So I'll just start by saying that I work primarily at the Broad Institute, which is a research institute that has a long history of doing genetic research in Boston. It's associated with MIT and Harvard. And we there at the Broad Institute, as well as centers really around the world, have been using sequencing technology to understand the genetic code of the SARS-CoV-2. And using that information to really understand what this little beast looks like at the most fundamental level. And from the very beginning of COVID-19, as early as I think the first week of January, when the alarm was really sounded about something interesting and potentially dangerous happening in Wuhan, one of the very first things that scientists did was isolate the virus and sequence its genetic code. And it was on that basis that, in fact, we identified that the microbe, the pathogen that was causing this strange illness, was in fact a coronavirus. It's something that we as scientists are familiar with, but we had never seen this particular coronavirus before. And so after a bit more analysis, it emerged that it was a new member of the SARS coronavirus family, similar to the virus that caused the SARS-CoV outbreak, quite a while ago now, but that we've seen before in Canada and around the world. But this was a kind of a new cousin of that virus, and one that we had never characterized before. So everything that we know about it, we've really learned in it's remarkable. COVID time, it feels like much longer, but it's really only been a few months. And so we're just really starting to understand what we're dealing with here, really in all aspects. And so with using genetic sequencing first to identify the virus, we continue to sequence virus samples that are collected from infected patients typically. We call those isolates because they're isolated from affected gen. And by sequencing, looking at the genetic code of those viruses, we can track whether the virus is changing or mutating as it moves from person to person and country to country over time and around the world. And so my work and the work of many others is to collect that information in real time and to analyze what is changing in the virus and what that means, if anything. And then also to use the information to understand how it's spreading.
So then I guess I would say,
Mm-hmm.
I've heard people talking about there's X number of strains now. And, there's some
Mm-hmm.
articles about these are the 8 strains circulating around
Yeah.
the globe. And then now there's 42 strains. And, so I guess,
Right. It's
how would you answer that question of, how many strains are
strain,
there
the
and how
strain
different are
question.
they?
Yeah. And the question about mutations.
Mm-hmm.
Mutations in a virus and what do they mean? So I'll start with the simple answer to the number of strains question. The answer is that in fact there's only one SARS-CoV-2 strain. There are many reports that I think are just misguided and potentially preliminary at this point, but as we know right now that are just wrong. That there are multiple strains circulating. There's only one strain. There are indeed mutations that are emerging in the virus's genome and that are being spread naturally. And that's a very normal thing. And mutations aren't inherently bad at all. They just happen as a natural process of as the virus replicates and makes copies of itself. There are little errors that get introduced into the genetic code. And by and large, those have no consequence on the kind of the biology and the important features of the virus in terms of how it infects and causes diseases in humans. So how it transmits, how it causes illness. They're really kind of silent, benign changes in the sequence. And so there are many of those, but none of them have emerged as a mutation that actually changes the biological, the kind of functional, a functional property of the virus. And that is really, I think, the definition of a new strain. When a mutation that emerges and that becomes, to some degree entrenched in the population, actually changes something functional about maybe the way that the virus transmits or how it enters, to infect cells or other features of the virus, the biology of the virus. So there aren't any of those.
So what would you call those different versions?
Actually, that's a good question, and I hesitate a little bit because even I think in the viral genomics, viral kind of evolutionary biology community, we use these words loosely, and sometimes we may be contributing to the problem of misunderstanding here, because sometimes the word strain slips out in a kind of more conventional colloquial way rather than in a biological definition. But I think you would call those different lineages. So if you could see that particular viruses with certain genetic features that don't change the function, but just kind of give a set of viruses its fingerprint, its genetic fingerprint, you can identify those as lineages. You could just call them isolates. They're just individual isolates, I think, with unique genetic signatures. So I think isolates, sometimes we call them clades in a similar way, when there's kind of a group of viruses with a common genetic ancestry, that where you can see that certain mutations have been propagated through a certain population of the viruses. We would refer to that as a clade. But again, it's not a strain, and it's not the mutations aren't causing a more dangerous form of the virus, or a kind of Franken COVID, mutation that is something we need to be really worried about at this time. They're just natural signatures.
Yeah. So what sorts of analyses do you do on these different versions to get at that question of the functional relevance?
Functional. Yeah. So actually, I think there's a really a popular example of one of these mutations where the scientific community and certainly the media have picked up on and are really looking closely at a particular mutation in the what is known as the spike protein of the virus. It's one of the pieces of the virus that kind of stick off the surface and that are exposed to the environment. And it's actually a really important target because it's in the region where we would be focusing on making a vaccine. So people are really curious to see if this mutation does confer any kind of functional change to the virus. And you may have seen the way this is referred to is that it's a D614G mutation, and that means that an amino acid at the 614th position of the amino acid chain that makes up the virus has been mutated to from a D, one type of amino acid, to a G, another type. And the D was the original version that emerged in Wuhan and mostly circulated around Asia. And the G is called the derived or kind of the new version. And it mostly moved westward from Asia through Europe and into kind of the east coast of North America. And there's some, I would say, anecdotal evidence that this mutation, the G version, confers increased transmissibility of the virus, but that's really based on kind of observational data. There's no kind of hard science that shows that. It's just a good hypothesis at this time. And the way that first of all, getting at that is actually pretty tricky because it's hard to do experiments on SARS-CoV-2 either in the real world because there aren't many human volunteers who want to get infected with different versions to see whether in fact the virus transmits or causes diseases more easily. And it's also really hard to do this work in the lab because this is a very infectious and deadly virus. As we know, the experiments on the virus itself need to be done with full PPE and high containment, very specialized facilities. And so it's really actually quite tricky to manipulate it in the lab and ask these kinds of questions in a more controlled environment. But the way you would get at it is both by doing population, kind of human level population studies and lab-based kind of more mechanistic studies. In human populations, you would look at places where there's a mix of both the D and the G form of SARS-CoV-2 naturally circulating. And there are, in many cases, many regions of the world have predominantly one or the other, but some have a mix. And then given that you have, in the same, say within the same country, a mix of both types of virus, you could see if there's differences in the population in terms of how the virus seems to be transmitting or how it's causing disease, say disease severity. Is it increasing hospitalizations? Is it increasing death or other severe complications? And then in the lab, we look at ways of looking at how well it infects cells or what kind of damage it does to cells, in a petri dish, basically that kind of idea, and seeing if in a molecular experimental way, you could see evidence that the virus with this derived version and this kind of newer version of SARS-CoV-2 actually increases transmission or some kind of pathology in an experimental sense. Then you'd again want to go back and look at that in human populations to see if it's true there too. So those are, I'd say, the two ways you would get at that problem. But at this point, it's still very much a hypothesis, and there's really no substantial evidence that it will be a new strain.
So is that really the only one of all the different variants out there that even has a possibility of having a functional difference, and the rest you rule out based on computational or other reasons to think it's probably completely,
Yeah.
meaningless?
Uh, it's certainly the one that the research community and the medical community is watching the most closely. There are studies to look at whether other mutations correlate or associate with different observations in patients. Do we see mutations that are correlated with higher disease severity, or more severe disease, or with particular types of disease? So some people have more cardiac involvement, some people have more severe lung involvement, or just more severe death as an outcome. But I'm just trying to think if there's any other ones that are really kind of strong candidates. I don't think so at this point.
So the reason I was asking that question is because I think maybe you didn't touch on the way that we can easily rule out a lot of changes because they don't even change any proteins or
that's right. Yeah, that's right. Oh yeah, there are many genetic changes that don't actually change anything in the way the virus kind of assembles itself. They're silent mutations, and so we know that those ones aren't going to have an effect. We'd be focusing on the ones that potentially could. But I think the upshot, the take-home message really is that mutations are normal. In many cases, they're even a helpful thing because they help us to track the virus, which is a different kind of application of the genetic code. Another use of it, but and so far there's only one strain, and there's no real evidence for a new SARS-CoV-2 strain that is more transmissible or more pathogenic.
So what have we learned from all this change tracking about sort of the rate of change? And can you help put some context about comparing this virus to others?
Yeah. So the rate of change.
Would you consider it to be a rapidly evolving virus or an average sort of?
I see. Yeah. No, I would say on the spectrum, it's probably a more slowly evolving virus. But in terms of how long it takes for kind of a new mutation to emerge as it passes from person to person. But because there's so much of that passing from person to person, there is actually a lot of genetic diversity that's out there in the virus, but it actually accumulates, like in terms of the rate, more slowly within the genomes, within the virus.
Yeah, because there's, I guess people are always trying to tie this to vaccines, and does this mean we're never going to be able to have a universal vaccine, that we have to have a different vaccine for each one, and all of that?
I would say too soon to know, but compared to, for example, the seasonal flu, where we do know that we haven't yet achieved a universal vaccine, and we do need to create a new one each year based on trying to anticipate what flavor of the flu is going to spread each flu season. SARS-CoV-2 does change more slowly, and so there's some optimism that a universal vaccine is possible, but there are many other factors that make creating a vaccine challenging. And it's nice to know that we don't have to struggle as much with the mutation rate of this virus, but there are many other challenges. So we'll see. Yeah.
Okay. So let's move on to talking about transmission dynamics, I guess. So you've been involved in a lot of work on seeing how this virus is spreading and using the fingerprint to do that. So can you just tell sort of a little bit about what's being done and then what we've learned?
Right. Sure. Yeah. So this is, as I was mentioning, another use of studying the genetic code of SARS-CoV-2 or of any other bugs that cause disease, is that these mutations that happen mostly naturally give us a fingerprint of the virus or the viral kind of populations that are spreading through particular communities, and it allows us to track it's kind of like understanding the enemy at its most fundamental level, the genetic code that makes it up. It's like having its secret war plans in a way that help you to know kind of where it's come from, and gives you some indication of where the at-risk places might be that it could be going, and how it's seeding infections in different communities. Mm-hmm. So we and many others around the world are actively collecting samples, usually discarded when someone has a diagnostic test done. We get a residual bit of the discarded diagnostic material, which contains the virus. And we use that to sequence its genetic code from that sample. And then we take a little bit more information from the person who the sample came from, maybe like where that was collected, what hospital or clinic, and some sense of what date that was, and sometimes some other characteristics of that might be relevant about the kind of demographics. And then we can use the genetic information to understand where the virus. No, it's not really done at the personal level. We kind of look across people in groups at the population level to understand where the virus, say, in a particular community has come from. How was it introduced? Or how is it continuing to be introduced? Mm-hmm. So an example from that, so where I work is based in the Boston area. And so we have asked questions about how did SARS-CoV-2 arrive in Boston, and when it came, what could we learn from that might influence how we might think about future control measures? For example, did most of it come from international travel, or is most of it coming from New York City? So at that level, just understanding the dynamics and the kind of migration routes of the virus. And then at a smaller scale, we and others are working with like local hospitals or local facilities that have higher risk of infections, like nursing homes. A good example is just asking the question of whether there is transmission within those settings where the virus is spreading kind of within the walls of the facility, versus is it continually being brought in from the outside? Mm-hmm. And that might affect how you deal with the problem of infections happening in your facility, because if there's spread that's seeded from within, you're thinking about how to decontaminate a wing, or checking your ventilation. Whereas if it's coming in from people who are coming in from the outside repeatedly, say staff or visitors, you're thinking about making sure that they're wearing masks or using hand washing and sanitation, or restricting those introducctions. So it can help you to know where to direct your attention.
Yeah.
So those are just a couple of examples.
Yeah. Is there anything that sticks out at all about this virus? Again, putting this one in context, or is it a sort of plain vanilla virus, or is there anything sort of that's been surprising that people have found as they've followed these things? What have been some of the interesting
in terms
observations?
of the epidemiology?
Yeah.
Oh, there's all kinds that all kinds of things that are interesting about this one. I think every virus and every epidemic, or in this case pandemic, is its own own special beast, and we're only beginning to understand this one. I think one of the, some of the things are interesting, and some are interesting because we just don't know the answer yet. And I think one great question is understanding getting our head around what we really think the R naught is, the ability of this virus, of someone who is infected, to infect other people.
Mm-hmm.
And any kind of armchair epidemiologists out there have probably know a lot more about the, you know, kind of the epidemic curve and this metric of R naught than maybe they did 6 months ago. But there's been a lot of debate that in some settings we're seeing very low kind of transmission from individuals onward, and in other cases we're seeing very, very high, and the numbers can range from 1 or just above 1, 1.2, that kind of thing, up into the, you know, 4, 5, 6,
Mm-hmm.
and this is the number of individuals that a single infected individual then kind of spreads their infection onto, on average. And so, and in some instances, I mean, you know, you can see just rampant, rapid, really invasive spread throughout a population very quickly that would suggest R naught is very high. But then in other contexts, it doesn't seem to be transmitting as extensively, as rapidly. So I think that's when that will be interesting to converge on about this.
Is that sort of huge variation in R naught in different contexts normal, or is that itself unusual?
I think it's not abnormal, given that we know so little about this still. But I think nonetheless surprising that in different contexts we see such varying kind of potential for transmission. And there can be lots of confounding effects in terms of how vulnerable the population is. Is it, you know, in a nursing home setting versus in a you know, lower age group, and other factors. But I think we'll get there. It's just taking some time to put all the data together.
One of my takeaways from reading about this in the media and the scientific literature is that, you know, number one, the preventative measures can be extremely effective, right? I mean, that's great. That's great news. And also, just there's a huge amount of difference in the risk of different contexts. And I think that's all of us that's what we're all struggling with right now, is understanding what is the real risk in different situations so we can move on with life in some way safely.
Yeah.
So the last thing I want to talk to you about is diagnostics. It's, you know, it's an area that I think a lot of people don't understand. Why is this not more straightforward? Shouldn't we be able to test everyone? Why is that not happening? So maybe you can first tell us, what does a diagnostic test actually entail? And then we can get into why is it not straightforward and what are some of the barriers?
Right. The current gold standard for testing for COVID infection, for coronavirus, is a test that involves direct detection of the genetic material of the virus itself, and it's an assay. It's a test known to scientists as a qPCR test that involves amplification of a tiny bit of viral genetic material to the point that we can detect it using the technology we have. And then if we detect it, it's positive. If we don't, we call that a negative. But the thing that makes this challenging is that the kind of the system for doing that is actually pretty laborious and requires specialized machines, specialized chemical reagents, specialized staff to run the test and to analyze the test. It's not a black and white outcome. And so if you were to dream up your kind of dream scenario diagnostic test for a virus that is causing a global pandemic, I think this might be like the last one on your list of all the options that we currently have. It does work. It's just tricky, and it's nothing like some of the other things that we more conventionally think about when we think about a medical test where you just get a simple result, positive or negative, say like a pregnancy test, for example. And even some other simple like tests that exist for other infectious diseases. This one came on too quickly. We didn't have really a special kind of point of care, easily deployable, easily readable test ready to go. So everyone was kind of scrambling to put together something that was robust and reliable. In the diagnostic world, sensitive and specific are the criteria, but that could be scaled and rolled out really globally as we've seen the need.
I know one question that I wonder diagnostically is how confident can you be that you have the right answer when you get one? Or is it 100% or is it not 100%? And how
right.
how much confidence do we have in our results?
I think it's fair to say that a positive test is a positive. So the false positive rate is very low. If you get a positive test, you have the virus in that sample. If you get a negative result, that's where things get trickier. And there it depends on many factors. One is, might you have an infection, but just at a very low level? And we still don't understand actually the relationship between the amount of virus in your system and whether you have symptoms or whether you can transmit. And it also depends a bit on the prevalence in your community, I would say. So if you get a negative result in a place with very little COVID, you're probably negative. But if you get a negative result in a community that with a lot of transmission, especially if you're at high risk, if you know you've been exposed, or you think you might have been, or if you've been like active in the community because of your work, or for any other reason, and just are at higher risk, and for that reason you get tested and it's negative, I would probably not assume that that's right. But in general, I'd say a positive test is a positive and a negative test, it depends. Right.
Why are some countries doing so well? Like what are those that are testing the most really nailing, I guess?
Yeah. I think that's a bit of a different question. Doing this kind of testing at scale is hard. For lots of reasons. And it was especially hard in let's say in North America, at least in, February, March, just as this was coming online, as everyone scurried to stand up the capacity to do this complicated test that I was describing in their hospitals and in their clinics and in their, public health labs. And so it's just not nothing about it's not just the technical implementation of the test, but the whole system, the whole workflow, the whole supply chain, the whole kind of specialized training that's needed, the kind of workforce that's needed to do the testing and interpret it, needed to be spun up to do at scale, and the need just happened a lot faster than certainly in the US than we were able to do as the cases started really exploding in many areas. It does, as I mentioned, it requires specialized equipment, specialized chemical reagents, specialized workforce, and so putting each of those pieces together, not to mention collecting the sample itself, the swab, that the sample goes on, accessing the tube that you put that sample in, the liquid, they call it viral transport medium, that you stick the swab in, there were shortages of that. Then it's, getting it into the lab and then reading the results and knowing when you have a true result versus when you need to rerun to be sure. Just all aspects of it are not really this test, and the way that you implement it end to end is not designed for scale. We've, I think, kind of brute forced it to become the at scale option because we haven't had another option. And still don't, frankly. There are some things brewing that could supplement or potentially eventually replace the current test as the gold standard, but those are still in development.
Do you see a path forward to having the capacity we need? A lot of people talk about for ongoing, back to work, we need to have surveillance capacity. Do you see a path forward for that?
Yeah, I think actually it comes back to the one of the solutions to achieve that kind of scale comes back to genetics and genetic sequencing that we were talking about earlier. So there are, across Canada and North America and really around the world, there are many hundreds of thousands, I would say, of these machines that sequence the genetic code of anything, including the COVID, SARS-CoV-2, that have not really been put to the use of diagnostics. So when we sequence it, and when other groups who sequence these samples do this, we're not using that information to confirm or refute the initial diagnosis of that individual. We're using it for subsequent analysis of the genetic code of the virus. But we could actually use the presence of the genetic code of the virus in the sample itself as a readout of a positive test.
Right.
And of course, we need to think about, how exactly to fine tune that to make sure that we know when we have a positive and we know when we have a negative. But the ability to do that at scale is tremendous. And it involves completely different supply chains and even like workflows than the current large scale tests, which means that it could really supplement what's currently established. It could be additive rather than alternative to the capacity that we currently have. And there you could be looking at, it's still the capacity of these sequencing machines themselves to generate these diagnostic test results are, it really depends on the institution or how many machines you have, but we're talking thousands, tens of thousands, hundreds of thousands of tests a day. If everything else can line up to feed those through the system. So you need to be able to collect that many samples and have all the right, bits that you need to do that, and to get them to the centers that have these sequencing machines and to prepare them for the sequencing process and get them through. But in theory, the machines can handle just a tremendous kind of massive scale of testing. And I think if we can work out all the upstream bits to feed that many, samples collected from people through, that could be a very great addition to the toolkit.
I'd love to ask you to look in a crystal ball and see how this, outbreak is going to progress, because every outbreak has its own story. But I know the answer is nobody knows, but what would you say are some of the factors that will sort of determine which way this goes, I guess, and some like things we don't know and some of the things that,
Yeah. Well, I think the big question is about immunity in the population, and whether, how do we get to, first of all, if you've been exposed, are you immune?
Yeah.
And that's still an unknown. And I think we probably won't know the answer to that until we start to see this anticipated second wave in the fall, when, other respiratory infections, including the flu, start to circulate again, and including, SARS-CoV. too. Yeah, so I think that's one big unknown. And I think from a more kind of practical perspective, I mean, getting people back to work is essential. We have to resume some semblance of an economy in a way that doesn't accelerate infections to the point that it totally breaks our healthcare system. That's always been the thing we've been trying to manage here. And I think the key to getting to that point is increasing testing, as we just talked about, so that folks can go back to work with confidence that they're safe. And I think we really need to address getting kids back to school. I think even if workplaces open for a lot of families out there, if your kids are at home, it's hard for you to go back to work. It's hard to really restart the economy in earnest if kids are out of school. And that means not only that the kids need to be safe, but the teachers need to feel safe.
Yeah.
And we, I think we all kind of we're only beginning to understand really the how kids are both susceptible and vectors or can transmit infection. So that itself is still an unknown, but we do know that older adults are susceptible, and kids, could be bringing things from their families or from their environments into the school that put teachers at risk, and in an unfair way. And I think that's having testing and productive ways to get school systems back up and running will really help us move forward.
What do you think are the prospects of, really rapid testing that could allow decisions to be made in a short timeline? I don't know, what is the timeline right now, and what would be the best case scenario of what could happen?
So I would say the timeline right now is on the order of about 24 hours. So I think sometimes faster in some cases, but say that's about average, and that's for a positive test. So I think if you're tested and you are positive, you'll hear very quickly, hopefully within less than 24 hours, same day ideally, or next morning if it's later in the day. And if it's negative, it may take a little bit longer to let you know, just because obviously they're prioritizing that feedback to positive cases. I think the best case scenario is for home testing. I mean, I guess that's the ultimate dream scenario, that people never need to leave their home to find out if they're infected. They don't need to go to a clinic where they put themselves at risk of infection if in fact they're negative, or put others at risk of infection along their route if they're positive. And so I think the ideal is to be able to bring the test to the person, for the test to go directly, through the mail to people in their own homes, or through, courier service. We're definitely not there yet, though there's lots of kind of models for doing that, and things being tried at small scale. The next best thing would be a point of care test, like a finger test if there was a blood-based test, or something that could be done in a doctor's office or at a kind of urgent care clinic, without actually needing to go to see a doctor even, but like by a nurse or nurse practitioner. So we are waiting with bated breath for tests that can be rolled out at that scale, that we trust, that are validated, and where the entire system supports that. So for example, if we're doing at-home testing, we need really robust turnaround on delivery of those tests. Or we could do at-home collection and then bring the samples back to a central place to be tested. That would, maybe be the second best, so that the individual never needs to leave their house, but the test leaves the house, and they get a result quickly. Yeah, and so I think those are just a few flavors of things that are in development and that hopefully we'll have solutions for soon.
Yeah, that I think it's good to end there with a little bit of an optimistic note that there are some great things in development to help to keep this thing under
Yeah,
control by following it closely.
right. Yeah,
Yeah.
for sure.
Any
Yeah.
last comments, messages you would want to, sort of whether it's debunking something that popularly you see in the media that doesn't make a lot of sense from scientifically, or any just sort of bits
Hmm.
of advice you wanted to throw out there?
Well, I think the only thing that I would say is encourage people to check the sources of the information they're consuming. There's a lot of misinformation out there, and I think everyone is fascinated by what we're all going through together, and sometimes people with best intentions are conveying strong opinions that aren't really rooted in data, in science, in evidence. And yeah, I would say that there are also a lot of people who are working hard on evidence-based information and sharing that in ways that are accessible to the population at large. And so encourage everyone to track down some of those great sources and get your information from there. And try to
Do you have any specific favorite sources?
Wow. Well, I think there's I think Johns Hopkins in particular has done a tremendous of sharing information in a very accessible way about this. That's one of my favorites. And then being a scientist, I tend to follow kind of more scientific sources, maybe Nature or the Lancet, or some of the more credible sources that write more kind of publicly accessible versions of those stories, and then trusted media outlets. I by and large trust what I see on the CBC. I trust their fact finding. I trust BBC News for the most part, just in terms of trying to find some news sources that do by and large do the digging before they put information out there. It's not perfect, and this is such a moving target, and there's so much to synthesize, and those reporters themselves have to sift between fact and fiction. So nobody's perfect, but I do. And then on Twitter, there's some great voices out there. Mark Lipsitch is one of my personal favorites. But I think finding some and just if you're curious, just Google, do a little digging, a little background checking on your sources, and sometimes even that's interesting in and of itself. You can learn a lot about how information is engineered and shared in this world, and it's worth taking a moment to know about who you're listening to, let alone what they're saying.
Right. Yeah. One thing I would add on the media is even if it's from a trusted source, to always read the full article, well, the media in particular, because so often the headline really emphasizes the scary part, whereas if you read the full article, they tell you the whole story, and it's not as scary when you put it in context.
That's absolutely true. Yeah.
Like these.
And I know this is exactly what you're doing. So so of course.
Fueled by Science would be, I missed that on your list.
So. Yes, I should have said that first. But huge respect to what you're doing here. We need so much more of it. And so thank you.
Well, thank you for your time.
Oh, my pleasure.
Bronwyn, right. We took enough of your time. So thank you again, and keep at the amazing work that you're doing, and keep sharing with the rest of the world what you're finding.
You too. Yeah, great.
We'll be following. Okay.
Okay.
Take care.
Take care. Talk to you soon. Okay. Bye for now.
Bye.
Bye.